Alright, in the first part of this lecture you learned a little bit about the basic biology, phylogeny, and ecology of the Archaea. Now we're really going to move into talking about some of their molecular details and how some of them are similar to what is seen in bacteria, and in other ways they're similar to what is seen in eukaryotes. Like all organisms, like all cellular organisms, Archaea are faced with the challenge of fitting all of their chromosomal DNA into a single cell. So fitting a large piece of DNA into a very small area. I should mention that like the Archaea, or like the bacteria, Archaea have one chromosome, and it's usually circular. Okay, so that's, in that way they're like the bacteria. So the way that Archaea achieve DNA packaging, they do use supercoiling, as do all organisms, but what's really cool about how they achieve DNA packaging is that they use histones, actual histone proteins. So histones are small basic proteins that are commonly found in eukaryotes. So all eukaryotes use histone proteins, and they use them to help pack DNA. And many Archaea, instead of some, I should have said many there, also use histone proteins. Note that no bacteria use histone proteins. So what do these histone proteins do? They are small basic proteins, and here is an image of a histone dimer, two of the proteins associated together, which is a very common configuration for them. And you should be familiar with the histones from studying structures that are called nucleosomes. Over here on the right is a nucleosome. That is a cartoon of a eukaryotic nucleosome. So when you took genetics, we would have talked to you about nucleosomes. In eukaryotic nucleosomes, what we typically see are 8 histone proteins organized with double-stranded DNA coiled around them into structures referred to as nucleosomes. And what we see in Archaea, archaeal nucleosomes have 4 histones per nucleosome. Okay, that's what the Archaea have. So that's a little bit different, but it's still interesting that they have evolved to use the same kinds of proteins as what the eukaryotes do, rather than using the other types of proteins used by bacteria, and I won't get into those here. Another really interesting feature about Archaea, although this is in fact only true of hyperthermophilic bacteria, okay, those that love extremely high temperature, is that they use and make a protein called reverse DNA gyrase. We previously spoke in class about sort of regular DNA gyrase, which is a protein that's important in introducing what are called negative supercoils into DNA, and that's what's commonly seen in most cellular life. But in the hyperthermophilic Archaea, we actually see a very high level of positive supercoiling, which is supercoiling in the other direction. And those positive supercoils are introduced by the enzyme reverse DNA gyrase. And scientists are really intrigued by this because in the lab, when you knock out the ability to make reverse DNA gyrase, the cells are actually still viable. So it's not essential for cell viability to have this protein, but nonetheless, when you look at all of the hyperthermophilic Archaea and some of the thermophilic bacteria as well, what we see is the presence of this enzyme, and we see a lot of positive supercoiling. So scientists hypothesize that these positive supercoils may play a role in eliminating the unpaired DNA regions that might happen from high temperature causing breaking of the hydrogen bonds called base pairs that are connecting double-stranded DNA together. That there would be a tendency towards that in high temperatures, and that reverse DNA gyrase and the act of positive supercoiling might sort of fight back or combat that tendency. That's an hypothesis right now. It's not a proven theory, but nonetheless it is true that the hyperthermophiles all have reverse DNA gyrase and a high level of positive supercoiling. Let's think a little bit about how archaea replicate their chromosomes. So I mentioned earlier that archaea have one circular chromosome, much as is seen in most bacteria, but an interesting difference in the chromosomes between bacteria and archaea are the origins of replication. It turns out that bacteria, as you should know, typically have one origin of replication. So over here on the left, we have a bacteria or a bacterial chromosome, and we see one origin of replication, and we see bidirectional synthesis proceeding outward from that one origin of replication, and that would proceed all the way around the chromosome on each side, moving at the same rate until they met in the middle, and then from there you would get two chromosomes, and those would go off into their two respective daughter cells during binary fission. Now what you see when you look at archaea is rather than one origin of replication, we actually have multiple. And shown in this figure over on the right is a cartoon depicting that. So on the right we have an archaeal chromosome, and it has 1, 2, 3 origins of replication. So we're seeing bidirectional replication outwards from 3 different points on that archaeal chromosome. You would expect this replication to be faster, and I don't really know if that's true. I don't know if cell division is uniformly faster in the archaea. I don't think that it is, but nonetheless this is the strategy that they have evolved to do, and this is actually very similar to, you guessed it, eukaryotic chromosomes. Which have multiple origins of replication. So even though they're much tinier, and you wouldn't think that they would need so many origins of replication to do cell division quickly enough, this is what they have evolved to have, and this is a difference in the archaea relative to the bacteria, and a similarity between the archaea and the eukaryotes. Another thing worth mentioning is that the proteins, the many proteins that are involved in DNA replication in archaea tend to be basically simplified versions of the eukaryotic ones. For example, the major DNA polymerase that does DNA replication, the major DNA replication in archaea comes from the same family of DNA polymerases as the eukaryotic one, which is a different family than the DNA polymerase 3, which is the major DNA replicating enzyme seen in bacteria. So it seems that, in short, archaea have DNA replication proteins more similar to eukaryotic ones, albeit simplified versions of those proteins. There are a few differences in transcription in archaea versus bacteria, and ways in which the archaeal transcription processes are more like the eukaryotic transcription processes. One is in their promoters. So promoters, remember, are the DNA sequences that are found in front of genes to which regulatory proteins bind, helping to recruit RNA polymerase onto the promoter region and thereby initiating transcription. You should remember that in bacteria, it is a regulatory region of RNA polymerase itself called sigma factor that attaches to the promoter. And you should remember that in eukaryotes, it's a suite of proteins called transcription factors instead. So first let's just mention the promoters themselves. Archaeal promoters look like eukaryotic promoters. They look like simplified versions of eukaryotic promoters. And the major distinguishing quality that allows us to say that is the presence of what's called a TATA box, a TA-TA sequence. Over here we see it in an archaeal promoter, and we see the same thing in all eukaryotic promoters. And in another aspect pertaining to the initiation of transcription, transcription factors in archaea are much like simplified versions of eukaryotic transcription factors. So you'll remember when you were studying eukaryotic transcription that there were a suite of proteins. That cascade is shown over here on the right. There are a suite of transcription factors, the purple, orange, and green there, and the reddish color, those are all transcription factors. Their job is to associate in a particular order or sequence onto the promoter region, and after that they recruit RNA polymerase so that transcription can occur. So what you're looking at is initiation, the initiation sequence in eukaryotic transcription on the right, and over here on the left, you're looking at the initiation sequence involved in transcription of Archaea. We see the TBP, and we see TFB and TFE, and these are similar to transcription factors that the eukaryotes are using. Similar, but simpler. Same pattern again. The major enzyme that is responsible for transcription, or RNA synthesis, is RNA polymerase. So you'll remember that bacteria have one RNA polymerase, and eukaryotes have 3 RNA polymerases, and it turns out that Archaea have 1 RNA polymerase. So in that regard, in the number of polymerases, the Archaea are much like bacteria. In this image, though, you'll see a drawing, a 3-dimensional depiction of the major RNA polymerase in bacteria and Archaea. They, of course, only have 1, and in eukarya. And the one that is shown in eukarya is RNA polymerase 2. That is the RNA polymerase that's responsible for the transcription of most genes that are protein coding in eukaryotic cells. And if you look at this cartoon, it's pretty easy to see that the RNA polymerase that Archaea use is very similar to RNA polymerase 2 held by eukaryotes, and not very similar at all to the RNA polymerase that is made by bacteria. Now, of course, they all catalyze the same reaction, and they all have to bind to DNA, so of course there are similarities, but when you ask the question, what is the archaeal RNA polymerase most like, the answer is that it is most like eukaryotic polymerase, RNA polymerase 2. How Archaea terminate transcription is still an active area of research, but what is clear is that they don't use the same mechanisms that bacteria use. They use some oligo-T sequences, which are long stretches of thymine incorporated into the RNA to signal termination. And that's not so different from bacteria, but what's neat is that they don't seem to form hairpin loops. So hairpin loops are something that bacteria do, but they're not something that Archaea do. Okay? Similarly, the use of what are called Rho proteins, which are seen in bacteria, are not seen in the Archaea. So we know that they terminate transcription differently. They don't use hairpin loops. They don't use Rho proteins. So they are different in how they terminate transcription different in comparison to the bacteria. So a really interesting feature of archaeal genes is that although most archaeal genes don't have introns or intron-like spacing DNA, some of their transfer, should be a T there, transfer RNA genes and ribosomal RNA genes do have intron-like sequences. They process them quite differently than what eukaryotes do, in that they do not have what is called a spliceosome for the removal of intronic RNA. Instead, they do splicing using a very specific enzyme called an endoribonuclease. And that endoribonuclease is able to recognize splice junctions, cut at the splice junctions, that's what's seen down here, the result of that cutting, and then enzymatically ligate together the exon DNA, leaving a mature RNA transcript. And remember, this is seen only in tRNA and rRNA genes, so not messenger RNA as is seen in eukaryotes. Interestingly, if you look at eukaryotic genomes, the genes that we have for transfer RNA have very similar intron-like spacing DNA, and they do use a spliceosome rather than a specific endoribonuclease to remove that intronic RNA, but it does signal to the evolutionary connectiveness or relatedness between the Archaea and the eukaryotes. So that's a really neat difference. The bacteria don't show this pattern at all. So translation in Archaea is its own sort of animal. Now, Carl Woese discovered the Archaea by sequencing ribosomal RNA. So therefore sequencing, the RNA that is found in the ribosomes, and the ribosomes are what are responsible for translation. So he knew that the Archaea were not bacteria and not eukaryotes on the basis of those sequences, which tells us that, of course, there are some differences in how the ribosome is structured in an archaeon relative to a eukaryote or a bacterium. However, when you look at them from a larger perspective, the ribosomes of Archaea are actually very similar to bacteria. They have a large and a small subunit, as do all ribosomes, but they're the smaller size, the 70S size versus the 80S size. So this is what we see in bacteria and Archaea. Okay? So similarly, the number of riboproteins in bacteria and Archaea is very similar, and the identity of the ribosomal RNA. So both bacteria and Archaea have a 5S, a 23S, and a 50S piece or sequence of rRNA in their large subunit, and in their small subunit they have a 16S and a 30S. You are going to be sequencing that 16S piece of rRNA from your microbial isolate, just to point out some connectiveness to the lab here. In contrast, eukaryotic ribosomes are larger, and they have an 18S and a 40S in their small subunit. That's comparable to the 16 and 30 in a bacterial or archaeal cell. They also have one extra piece of ribosomal RNA. So they have the 28S and the 60S. Those are comparable to the 23 and the 50, but then they have the 5.8 and the 5S. These two pieces here, whereas bacteria only have the 5S. So that 5.8S is an extra piece of rRNA seen in eukaryotic cells. You know that the start codon is typically associated with a tRNA molecule that is called the initiator tRNA. That is true no matter what kind of organism you're talking about. You also should know that the initiator tRNA in eukaryotes is carrying the amino acid methionine, and in bacteria it's carrying the amino acid formylmethionine. So this is the bacterial formylmethionine shown here. It's got this extra formal functional group, and this is the regular old methionine amino acid seen in eukaryotes. And it turns out that it's also what is used by the Archaea at the level of their initiator tRNA. So the first amino acid in any polypeptide synthesized in an archaeal organism or a eukaryotic organism would be methionine, whereas in bacteria it would be this modified N-formylmethionine. So the take-home message here from this lecture, or one of the take-home messages, is that you should think of the Archaea very much as their own domain, because they are. They have their own biology, their own evolutionary process, their own ecology. You should know that we know, in fact, relatively little about the Archaea. We only have a few of their members in pure culture. A lot of our information about them is based entirely on sequence data collected from the environment, and you can do some kinds of experiments that way, but you can't really interrogate the physiology of an organism until you have it in pure culture in the lab. We really only discovered this domain in the late 70s, so that's not actually all that much time to be working on this. And also it's important to note that perhaps because they're so difficult to culture, but also probably in large part because there are no known archaeal pathogens, not a lot of research effort is focused at understanding the organisms that belong to this domain. I think that that will slowly, or I hope that that will slowly start to change as people start to think up applications for these organisms, for example, using their heat-adapted enzymes in the case of hyperthermophilic Archaea for different biotechnological purposes. Also, as we learn more about the human microbiome, People will become more interested in the methanogenic and perhaps other archaeons that are present in the human body. That should spark a better understanding of the organisms that belong to this domain. And all of that will provide insight into the evolution of life on Earth, because this is a very important group of organisms, and by better understanding them, we will better understand life on Earth. So this particular figure shown here is taken from the lab page for scientist Dr. Finn Werner. He's a faculty member at University College of London in the UK, and he's very interested in the archaea and in archaeal molecular biology and in evolution. So this figure shows how the RNA polymerases, which are designated RNAP here, as well as a universal elongation factor that is important in transcription, and that universal factor is shown as SPT5 here. He's showing how these evolved with the first cells on Earth, and that is why all the domains have RNA polymerase and that particular universal elongation factor. And then he's showing how sigma factor is unique to the bacteria, while the archaea and the eukaryotes share transcription factors. So that was something I covered earlier in this lecture. Also, please note that the eukaryotes have evolved to have some additional transcription factors that are not shown here. So people like Finn Werner are busily at work securing grants and funding and doing good research, looking at better understanding this domain, and I think that there might be some really interesting future projects for any of you that might want to go on in microbiology in this particular area.